Welcome to the Anxiety Summit 6, Toxins, Medications, Infections. I'm your host, Trudy Scott, food mood expert, certified nutritionist, author of the Anti-Anxiety Food Solution. Today we have Dr. Cara Fitzgerald here to do a deep dive into methylation, and we're going to talk about the psychiatric implications, toxin connections, and how methylation impacts immunity. We'll also cover powerful evidence-based diet and nutrition and lifestyle approaches to support methylation. Cara Fitzgerald received her Doctor of Naturopathic Medicine degree from the National University of Natural Medicine in Portland, Oregon. She completed the first Council on Naturopathic Medicine accredited postdoctorate position in nutritional biochemistry and laboratory science at Metametrics Clinical Lab, now Genova Diagnostics, under the direction of Richard Lord, PhD. Her residency was completed at Progressive Medical Center, a large integrative medical practice in Atlanta, Georgia. Dr. Fitzgerald is the lead author and editor of Case Studies in Integrative and Functional Medicine and is a contributing author to Laboratory Evaluations for Integrative and Functional Medicine and the Institute for Functional Medicine's textbook on functional medicine. Most recently, with Romilly Hodges, she authored the ebook, The Methylation Diet and Lifestyle. Dr. Fitzgerald is actively engaged in clinical research on the methylome using diet and lifestyle interventions in conjunction with Health Gut Research Institute. She's a research clinician for the Institute for Therapeutic Discovery. Dr. Fitzgerald is on the faculty at IFM, is an IFM certified practitioner, and lectures globally on functional medicine. She runs a clinical development program for professionals and maintains an active blog and podcast series on her website, drcarafitzgerald.com. Her clinical practice is in Sandy Hook, Connecticut. A very big welcome, Dr. Fitzgerald. I've got so much respect for you and the many hats you juggle: clinician, researcher, writer, podcaster, mentor to other practitioners, and of course, as a methylation expert. And I'm just so pleased to have you here today to share your expertise.
Thank you. It's a privilege. Thanks for asking me, Trudy.
Well, thank you. You like to say that the dance of methylation extends far beyond supplementing with B12 and methylated folate. So tell us what methylation is and what do you mean by this?
Yeah. So methylation, so a methyl group is a carbon and 3 hydrogens. It's just outrageously fundamental. We evolved with an abundance of carbon and hydrogen in the environment, and, just economically, it made sense for us to, use them all over the place. It's from what we, it's from the stars. We're from stardust, and we built our being based on these. And so methylation as a process, either making this carbon and 3 hydrogen structure and using it. So making it, adding it onto compounds, removing it from compounds, those 3 steps are vital for life, and they happen in the background in virtually every cell all of the time for just, myriad mechanisms. In fact, we're still identifying what we call the human transferase methyltransferome, all of the methyltransferase enzymes, we're still characterizing them. We just identified a mitochondrial DNA methyltransferase enzyme. We're unpacking the extent. And so, for folks not tuned to biochemistry, it's almost analogous to how we think about oxygen. We don't think about oxygen. So methylation is happening in all of our cells all of the time for myriad processes. And, kind of akin to how we breathe in oxygen, we don't think about it sort of, traveling down to a mitochondria where we're pulling electrons off and et cetera, and, moving it around on ferritin and on iron and red blood cells and so on and so forth. So it's this background activity that's just happening all over the place. And, the dance of methylation, so So we tend to simplify our thoughts around methylation, limiting it to the methylation cycle where we're taking B12 and folate or maybe betaine, sometimes the minerals associated with it, basically to help produce S-adenosylmethionine, which is the compound that the methyl group gets, ushered around our body on. So that tends to be where we put our focus. But in fact, if we zoom out beyond that, there are many non-methylation cycle ways in which we can we absolutely need to do to support methylation. So my area of research, as you mentioned in your introduction, has been looking at looking at DNA methylation. So the methylation that's involved in regulating genetic expression. That's been where I've kind of set my brain these last number of years. And we see that DNA methylation activity is impacted in ways far, far outside of just simply pushing methylation forward with those nutrients. So, for instance, as you mentioned earlier, toxins can influence DNA methylation. Toxins of all stripes, be it, reactive oxygen species, be it, chemicals, pesticides, et cetera, the whole gamut of them can wreak havoc, can cause imbalanced methylation. We know that stress can deplete methyl donors and directly actually impact how we methylate, whether we methylate balanced on our DNA or not. And let me back up and talk about why this is so important. We use methyl groups on DNA to turn genes off. So when there are methyl groups on a particular region of the gene, it will turn that gene off from activity. And, the opposite is true to turn the gene on. So when we remove methyl groups, or if a gene is not methylated, that gene is on. So toxins can cause something that we call aberrant methylation. It can lead to regions on the genome that are hypermethylated or hypomethylated. They're aging, actually. I've been focused a lot on the epigenetics around aging, and aging itself is a journey towards imbalanced methylation. So you can start to see really important genes that we want on, become hypermethylated in the aging process, and they're turned off. And particularly the most research in this arena is around tumor suppressor genes, the cancer surveillance genes, the ones that, as the name implies, suppress cancer. And so that's why cancer is a aging, by and large, an aging phenomena. We also see the same kinds of imbalances occurring, although there is less research around them, in all of the chronic diseases of aging. So we see that happening similar to toxic exposures. So not just methyl donors, but stress, toxins, sleep, exercise, we can cast a very wide net and see that all of these activities and more will impact whether we're methylating in a balanced way or not. So that's really what I'm referring to when I talk about the dance of methylation. It's so much greater, than B12 and folate. And, I would argue that we need to be mindful about overusing the isolated vitamins.
I'd like you to talk about that because I know you talk about oversupplementing, and that can actually cause increased anxiety in some people.
So the reason that I got into the whole exploration of the DNA methylome is because, again, going to cancer epigenetics, that's where the lion's share of science exists. So when I started to do a drill down into that literature in around 2013, what I saw was that the tumor microenvironment can hijack our epigenetic machinery, those DNA methyltransferase enzymes and the other players, and turn off tumor suppressor genes. So the cancer microenvironment itself can cause hypermethylation, shut down tumor suppressor genes, and hypomethylation on oncogenes, which promote cancer. So they get the tumor microenvironment can go in there and just really wreak havoc on our epigenetics. And so the question for me as a doctor became, am I, by supplementing my patients with methyl donors, could I, in certain instances, be promoting cancer? And the literature suggests, now this is new, but the literature suggests that in certain populations, yes. And we need to be mindful. So let me just finish this because I don't want to cause a lot of anxiety in folks. If we don't have enough methyl donors, we can actually increase our risk of cancer. And by, conversely, excess methyl donors in certain populations, particularly folks who are older, you may increase risk of cancer. So there's a U-curve relationship there. It's not cut and dry. I'm not going to advise people to stop their vitamins. I'm going to advise them to work with their doctors and, consider taking a food forward approach like we do. Now, thinking about anxiety, yes, yes. So I had this aha in 2013. Romilly Hodges, my director of nutrition, and I created our methylation diet and lifestyle program, and we started to use it in with our patients, and then later we researched it. But my very, very first patient I used this on was somebody who could not tolerate any methyl donor. It caused, actually in him, it caused a profound pain response, and secondarily anxiety. Plenty of patients have come into my practice. They're a minority, but plenty of folks have come in who can't tolerate methyl donors of really any kind. We'll see an increased anxiety presentation. Actually, I'm thinking back to when I was in the lab. The first time I saw this, I was consulting on a case at the lab of a woman with a history of, pretty severe panic disorder. And when she started methyl donors, she became agoraphobic for that period of time until we realized, she clearly needed to be off them. Probably one of the mechanisms around that is an inability to clear adrenaline. So the COMT mutation might be a piece of that intolerance to methyl donors. There are other reasons one might have increased anxiety, but that certainly was what I saw in this very interesting first case where she became agoraphobic. And we needed to do at that time, I started to flesh out this whole backdoor methylation idea where, maybe if we preserve methyl donors, if we preserve our S-adenosylmethionine, the universal compound that ushers the methyl group around to the body, if we preserve that, we won't need to lean on B12 and folate, et cetera, as much, and therefore we won't turn the problem symptoms on. And so, Trudy, I'm going to pop quiz you only because my teacher did this to me all the time. Do you know the two areas that demand the most S-adenosylmethionine, the two pathways that are the most methyl donor demanding? It's creatine, so it's muscle. It's maintaining your creatine, one of your main energy pathways, creatine synthesis and metabolism, and making phosphogroups, making phosphatidylcholine and so forth. So those are big demands. And so we can do a little bit of backdoor methylation in some cases by giving a little bit of creatine. We can preserve the body's need to make it by a little bit of supplemental creatine. So we may not be focused on building muscle in a particular individual. We're just kind of focused on trying to save methyl groups. And likewise, we can do a little bit of phosphatidylcholine. And that also will reduce demand on the body. So it takes 3 moles of S-adenosylmethionine, 3 different steps of SAMe to make it. So if we give a little phosphatidylcholine, or we could use eggs in somebody who can handle eggs, and that's my experience is that can be much better tolerated, and it can preserve total body methyl donor status, and you can achieve good clinical outcome and avoid doing the B12 and folate, or at least reduce it.
Okay, so for someone who doesn't have got no idea what a methyl donor is, that's the B12, the folate, the betaine, any of these things that are going to enhance methylation.
Yeah, those are the compounds those are the nutrients, the ingredients that we need to make our universal methyl donor S-adenosylmethionine. And then S-adenosylmethionine, or simply SAM or SAMe, will go around and take that methyl group and put it on, or, do all sorts of metabolic activities. Yeah.
Got it. Thank you. So, is this the fact that some people have an issue with these methyl donors? Could it be dose specific, or is it? You mentioned, there's many possible ways. It might be the COMT polymorphism. Could it be dose specific, that some people just need very, very tiny amounts?
Yep, absolutely. And there are different forms of B12. There are different forms of folate that a clinician can work with. You might even go topical. I've certainly used topical in very sensitive individuals, sometimes with a good result. Obviously, we're not going to be absorbing much of that, but it's worth a shot if you've got a particularly sensitive individual. You could do micro amounts sublingually. There are intranasal B12 sprays. You can use suppositories. Some people use IV or injection, but obviously we're going to avoid that in very sensitive people. But my point is that there are many routes of delivery, and there are different forms of B12 and folate. So if we need to use them, we've got a lot of options in the supplement world. And our program is food forward, so you can eat your methyl donors as well.
And we're going to talk about that in a second.
Yep.
So I wanted to talk a little bit about how methylation ties into mental health, and then also talk about detox. You mentioned how these toxins actually affect methylation, and then we also know methylation helps with detox. Let's maybe talk about the toxins first, because you did already mention the toxins.
Sure. Okay. So toxins gunk up the methylation works via just a broad sweep of mechanisms. When we think about genetics, toxins can directly, obviously, damage DNA. They can directly interfere with methylation, and then they can also interfere with the enzymes themselves. So they can, via a variety of mechanisms, they're problematic in the genome. And then when we look at the methylation cycle, so where we use the B12, folate, betaine, and make S-adenosylmethionine, so that cycle by design, it interfaces with the sulfuration pathway, which makes glutathione. That's a big player there. So they're intimately related, and under conditions of increased oxidative stress, where we need more glutathione to quench whatever's happening, the methylation enzymes are turned down automatically. It's just the nature of the beast, because we're going to direct. So homocysteine sort of is the switch in the middle. So homocysteine will get recycled towards making S-adenosylmethionine, or homocysteine will be shuttled down to make glutathione. So under conditions of oxidative stress, we take that homocysteine and use it to make the cysteine on the homocysteine, and use it to make glutathione. So it's a sulfur compound. Cysteine is a sulfur compound. Glutathione is a sulfur compound. And sulfur is just pretty brilliant at being able to bind and pull out all sorts of toxins, as well as quench free radicals. And glutathione is our universal sort of detox antioxidant player. So under conditions of oxidative stress, / toxin exposure, we'll shut down methylation. And that's by design, and it should be short term. We clean up the problem, and then we get on with life. By design. Unfortunately, we're living now, most of us, in, a relative toxic soup. So toxins can profoundly interfere with the methylation cycle and really grab up all of the cysteine to produce glutathione. Taurine is also made in that same cycle. Sulfate, so for sulfation, another phase 2 detox pathway, is made in that same cycle. So if we're under constant toxic burden, we're going to see some problems happening there. And so one of the easiest, to summarize, like another backdoor methylation step would be just cleaning up the toxins, like really simply.
So avoiding pesticides, fertilizers, BPA, phthalates, anything, those types of things.
Yeah, metallotoxins, mercury, arsenic, lead, etc.
Mycotoxins from mold.
Yeah, any glutathione demanding process. Taking care of mitochondrial health, one of the greatest use of glutathione is in quenching free radicals in the mitochondria. Actually, that trumps, toxin exposures itself. So just in the course of our daily life, mitochondria produce loads of free radicals just in the process of making ATP, and glutathione is used there extensively. So if you want your mitochondria in tip-top shape, and you want that whole antioxidant system supported, and that will preserve glutathione and, by extension, preserve methylation as well. So mitochondrial health is very essential.
Right. And then the fact that we are exposed to the toxins, now it's affecting our methylation, that could in essence contribute to mental health issues. But we also have the MTHFR polymorphisms, which research is showing are associated with psychiatric diseases. The paper that I wanted to mention is methylene tetrahydrofolate reductase in psychiatric diseases, which is a paper published in 2018, and they talk about anxiety, depression, ADHD, schizophrenia, autism, and bipolar disorder. And they actually also talk about one of the genes, the C677T, being associated with postmenopausal depression and childhood trauma related to major depression disorder. So I'd love you to just talk a little bit about this connection with methylation and mental health.
Yeah, it's a good survey. It's actually a really good review, and that's Lee Wan et al. In Translational Psychiatry. For folks who want to access the paper, it's open access. So certainly the 677 polymorphism is going to impact methylation greater than the 1298. I guess what I want to say about it, Trudy, is the 677 is so common. It doesn't impact mental health in everybody, and it's definitely something to consider. It's an important player, and it can be a game changer, but it's not a death sentence or a life sentence either. So I think it's important to understand that about it. It's going to slow down the methylation cycle. It's not going to grind it to a halt. If it did, you wouldn't survive, because methylation, as I started our conversation out about, is used all of the time everywhere. So if you're not able to methylate, you don't get out of embryogenesis, you just don't survive. Probably people who do better with the 677 mutation are doing, unknowingly have some of these backdoor methylation activities happening, like perhaps their toxic burden is less, maybe they're exercising, sleeping a little bit better, et cetera, eating methyl donors in their diet. So there's a lot of backdoor ways we can tend to it. And the degree that the mutation is inhibiting methylation varies, but it is a significant player. So the mutation itself is a significant player. And what's also interesting is that the gene can become hypermethylated. So even without the mutation, you can have abnormal DNA methylation patterns and inhibit expression that way. So basically what it means is that we need to be thinking about methylation in all of our patients. and evaluating for functionality, regardless of whether or not they have mutations. And we need to understand that, not every mutation is equal. Not everybody needs to worry about it. Not everybody needs to be on high dose folate and B12. But all of us need to be optimizing our methylation. And, again, as we'll talk about, doing a food forward approach, I think is is the safest way to start. And then we can layer in nutrients, isolated vitamins later if we need to.
Okay, so it's very interesting that you mentioned this, that we need to be thinking about methylation in all our patients and clients, and you may have the polymorphism and it may not be causing a problem, and you may not have the polymorphism and you may still be having a problem. And,
Yes.
I mentioned this when we were getting ready for the interview. A lot of people, you'll see this on social media and in email saying, I've been diagnosed with MTHFR, what should I do? So can you just clear that up?
I mean, I have had patients. I actually had a woman come to see me with pretty significant abnormal findings associated with. Well, she needed a work. So her blood work came back very suspicious of a potential leukemia, and we needed to follow up with that. And her focus was not on that. Her focus was on her MTHFR mutation. She had locked onto it like so many people. She's kind of the extreme example. And it was very challenging for me to put perspective on that MTHFR and get her to go to a hematology oncologist for workup on her blood findings, which she absolutely needed to do. And I know some of your folks out there in your audience are thinking that maybe MTHFR had something to do with that. And yes, it's possible, but it's also, we don't know that. And certainly addressing the MTHFR with B vitamins would not be appropriate to address the changes in her blood work. So yeah, plenty of folks come to us with just huge anxiety around their single nucleotide polymorphisms, and MTHFR is the biggest one. You can go into the literature and you can look in some pretty good journals where they absolutely dispute the validity of the mutation. But then you can also you can also look in plenty of other good journals like the Translational Psychiatry article you just mentioned and see that it is relevant. So my position is that it is relevant. We do need to look at it, and we also really need to understand that it's not a deal breaker, and it doesn't. It should be something that we consider in the overall context. Using a full systems or functional approach. It's a piece of the puzzle. There's a continuum. So for some people, it is not a big deal, and for others, it's a big deal, and we need to address it, and we need to address it strongly. So we need to find out where you are on the continuum. All the variables need to be addressed for optimal health, and that's but one.
Thank you for clarifying that. And I must just apologize because I didn't mean to laugh when I said that, because as you said, there are a lot of people who are very anxious about it, very concerned about it, and we do need to take their concerns seriously.
Yeah.
So if anyone...
Well, I understand your chuckle. It's a chuckle that I've had as well. It is because we see it so much, but this particular individual is always top of mind for me because it was so... challenging. So there's so much discussion online, and I think misinformation, a lot of fear, and folks who are using Dr. Google can latch onto it in a way that is not balanced and not helpful for overall well-being.
Right. But the encouraging thing is, and you're going to talk about this now, is that there's diet and lifestyle tools that you have that are very effective, they're safe, and they're research-based. And this is what you use in your practice. So let's talk a little bit about some of this very simple but very effective approaches.
Yeah. So first, I want to remind people, as we were talking about toxins, toxins will definitely influence methylation negatively. So that's a low-hanging fruit we can all be working on to the best of our ability. Getting adequate sleep. So we know that insufficient sleep causes, again, aberrant methylation. I'm talking about DNA methylation here. So sleep, exercise. Exercise favorably impacts DNA methylation. It also increases it turns on the methylation cycle, and we can see improvements in homocysteine and so forth. So exercise, smart exercise, not over exercising or pushing beyond your limits, but maybe like 60 to 80% perceived exertion. That's what we used in our study. Actually, you can push up against that sometimes as well, but in our study, that's what we decided the sweet spot was. Meditation. So stress, as Trudy is focusing a lot of her work on, will absolutely lead to imbalanced methylation. In fact, one of the areas that we're most interested in our study, and the first publication is going to be on aging, and we looked at a biological clock, a marker of aging, actually more sensitive in predicting mortality than age itself, than chronological age, and it's looking at methylation of the DNA. Specifically 353 CpG sites is the clock that we used. Those CpG sites are on the DNA where a methyl group is. I'm sorry, I don't want to get too granular here, but about 25% of the clock genes are associated with glucocorticoid activity in some direct and indirect way. So aging process itself seems to be pretty profoundly tied in to stress over time. We also know, if I don't forget, I think I'm veering off track here a little bit, but I want to say it, that our early life experiences, our in utero experiences, even past generational stress experience can impact how we respond to stress in our lives. So early, if you grew up in a stressful household early on in your life, very early on, that can actually make you more vulnerable to having to your stress response jumping on. And conversely, if you grew up in an environment where you received a lot of comfort and connection, that can actually the reverse. You can have more stress resilience. So those of us that are many of us have had a high stress experience early on. We need to just put attention there. It's not a life sentence or death sentence. We can shift it by using certain tools like meditation and so forth. So there's all these important backdoor activities. But then there's the diet. Methyl donor foods, there's plenty of them. Of course, most commonly are greens. We want to ingest our greens as much as we can. If you're an egg tolerant, eggs are a beautiful methylation superfood. Liver, if you're open to eating some liver, clean source liver. In our study, I have to say I don't know, maybe half of the people were interested in consuming liver. Most folks aren't particularly keen on it. I'll do a little bit of liverwurst from time to time, or do a little bit of pate, but that's a methylation superfood. Nuts, seeds, pumpkin seeds, sunflower seeds, sesame seeds, methylation superfoods. So our diet is loaded in methylation superfoods. There is nothing in the literature negative. There are no negative outcomes using these kind of dietary interventions. There's no increased risk of cancer like we've seen in some isolated vitamin studies. We see benefit really by, when you research these particular nutrients, we only see benefit. So these are methyl donor rich nutrients. The other area that we're very interested in are what we're calling methylation adaptogens. So it turns out there are many nutrients, mostly flavonoids, that impact the enzymes, DNA methyltransferase, and another one that cleans up DNA methylation marks. they're both families of enzymes, so there's a variety of them. So the DNA methyltransferases and the 1011 translocation enzymes can be nutrient regulated. Green tea, curcumin, luteolin, lutein from tomatoes, rosmarinic acid in rosemary. What else? Diindole methane, so DIM. We think about that for estrogen metabolism, but it's actually an epigenetic DNA methylation regulator, and there's some really cool research on these compounds. Really, really cool research. A lot of these compounds have been used time immemorial. You know, green tea has been used forever in traditional medicine. Curcumin certainly has a long use history. There are clinical trials using these nutrients in different conditions with good outcome. They're pleiotropic. There's many different ways that they work. My suspicion is, as we do more and more research on the epigenome and natural products, we'll see that their heavy lifting is here in their ability to kind of regulate the DNA methylation patterns, so correct poor patterns. Famously, again, cancer is where a lot of the science has taken place, and we can see that these flavonoids can allow for the re-expression of previously hypermethylated tumor suppressor genes. It's just so cool. Blueberries, you know, berries of all forms. I'm just like, in my daily existence, I'm always thinking about what methylation adaptogens am I ingesting, and, you know, my family, my daughter, my patients, along with having a methyl donor-rich diet as well, and the lifestyle components.
So I've got a few questions here for you, please. I wanted to go back to, firstly say I love livers, so I'm glad that you mentioned liver.
Oh, you do? Good.
Yes, I do. So
you're one of seven people in the world.
So I'm glad you mentioned that. But I wanted to just make a comment about some of these foods, because we do have someone in the summer talking about oxalate issues, and a lot of the foods you mentioned, the beets, the greens, and some of the nuts and seeds are high in oxalate. So this would need to be adjusted according to someone's specific dietary needs, I assume. And any comments?
Yeah. That's right. That's right. You would need to be mindful about that. Yeah, you know, beets are a superfood, sure. Yeah, I mean, if somebody, so the classic oxalate issue, of course, is kidney stones, and you want to be mindful around that. The research is a little bit equivocal there, but we want to err on the side of caution, and the diet is it's very adjustable. So the way that we've used it in clinic practice, which has been a lot of fun, is we layer the methylation diet and lifestyle principles onto whatever our foundational food plan is with a given individual. So if we've got somebody with small intestinal bacterial overgrowth, and we're going to be putting them on a FODMAP, you can layer in MDL principles. You can layer in MDL foods. If people are on a hypoallergenic or elimination diet, or a autoimmune paleo, or a ketogenic diet, or a time restricted or intermittent fasting program, the MDL principles are imminently layerable.
What does MDL stand for?
Methylation diet and lifestyle.
Okay, got it. Okay, good.
Yep.
And then the what else was I going to ask? Oh, tell me, what do you mean by methylation adaptogen? Because we hear the term, we know what the adaptogen is when we think about
it's a little
herbs. So just
yeah. So we're using it a little bit differently. The methylation adaptogen nutrients, it help. I mean, I guess we're using it a little bit classically, but the methylation adaptogen nutrients inhibit DNA methyltransferase activity, but they appear to inhibit it in sort of a selective way. So in vitro and animal research, primarily this is where it's done. Although we are looking at this in our study. I don't have anything to report on it yet, though. Tumor suppressor genes are hypermethylated and turned off, and these flavonoids appear to be able to allow re-expression. Okay. So they inhibit the DNA here. And when we think about, demethylating agents are used in oncology. There are demethylating drugs. They're hard-hitting, they're broad spectrum, they just shut off methylation. And so you can imagine they're associated with all sorts of different side effects. It seems like these adaptogens are selective. And when you use those inhibitors with the methyl donors, so we know that we need to support healthy methylation. It's happening all of the time. And we also want to direct methylation so that in the context of DNA methylation, it's happening where it should be. It's happening on the right genes. And so the combination of these flavonoids along with a methyl donor rich diet, these together appear to be able to allow us to find the sweet spot. And that's what we showed in our study, interestingly enough. So we were able to, I can talk about the epigenetic clock. We were able to, in our research group and not in the control group, we were able to kind of move around where methylation was happening. So we didn't increase net methylation in the study group, but we moved around where methylation was happening on the DNA, which is cool. And we moved it around in a way that actually made them younger.
Wow. How did you measure that?
We measured the DNA methylome. So we used a test available for research purposes only by a company called Illumina. This is not available to the general public yet, but it will be in time. We'll start to use these, or at least clinicians will be using them. There are a couple of proprietary aging clocks available. There's a lab called Zymo. They have a kit called My DNA Age, and that's available direct to consumer. It's a proprietary clock that they created at their lab.
Okay. Very interesting. So this is
really interesting.
Is this the study that is in progress now? Is this another study of yours?
We actually we finished it. We finished this study late last year, and we've been analyzing our data and writing it up for publication. And I'm actually writing this program, this methylation diet and lifestyle program I'm writing it up as a book for regular people, so people will be able to actually do this program and understand it and understand why we're interested and what we found in our research, et cetera. We did incidentally increase circulating folate without giving any vitamins, any folate. So we were able to do that through diet, and we haven't talked about this yet, but our gut bugs, our microbiome can make folate among other nutrients, and we gave a probiotic. So our study included for supplements a probiotic, a Lactobacillus plantarum, and then we included a green superfood, so a greens powder flavonoid-rich superfood, but no B vitamins.
That's amazing. Pretty amazing.
Thanks. It's really exciting.
Congratulations. So tell us a little bit more about the gut and the microbiome and the creation.
Yeah, I should have brought this up earlier.
No, it's okay.
I guess the good news is there's so many ways we can influence methylation. There's just so many ways that are outside of the box. Like, you don't have to run to the myopic, high dose B vitamins. There's many things that we can do, and I would argue that we really have to do to keep it balanced and humming along in an optimal way. So gut bugs regulate methylation by a lot of different mechanisms. The one thing that we were focused on in our study was, could we give folks Lactobacillus plantarum, and would that help potentially increase folate production? So in the presence of PABA, which we can get readily in the diet, and Lactobacillus plantarum can make folate. And we noticed that we increased methylfolate in circulation in our study participants. Now, is that coming from the Lactobacillus plantarum? Is that coming from the foods that they were eating? We don't know the answer, because we used a full systems intervention. We didn't use, just single interventions. So we can't say for sure, but we were able to do it. So we want to tend to our microbiome, and think that and know that tending to gut health is tending to epigenetic health is tending to methylation health. Now, interestingly, in a really dysbiotic gut, you can actually make way too much methyl donor. And you can, some clinicians out there listening to this talk, you've measured B12 and it's crazy high in someone and you don't know why because you're not giving any. I know sometimes we think that that's because there's, no intracellular uptake in B12. If that were the case, then you should see a methylmalonic acid elevated as well. And so I would test for that in urine. I wouldn't use the standard reference labs because they generally set the reference range to 3 standard deviations, so you don't see positives in blood. But if you do urine through a specialty lab, it's at 2 standard deviations. So if there's poor intracellular B12, you should see a high MMA, and you might see a high serum. So that can help you confirm or rule out. But our gut bugs can make, inactive B12 isomers, and they can also, make folate excessively. So you can have an imbalance in the folate producers, and then you could see high circulating folate in somebody who's not supplementing. And that could be a marker of, an imbalance. So it's kind of interesting that we want to we want our gut bugs to be making sufficient biotin and, helping with absorption, et cetera, and the other B vitamins, but we don't want them to overdo it.
And then, of course, when we've got, we no longer have dysbiosis and we've got a good gut, then we're making our neurotransmitters as well, which is going to help us with mood as well. So
absolutely.
Huge.
That's right. That's right. And you're actually going to preserve methyl donors in that way because they're produced locally in the GI and, not de novo in circulation.
Right. Now, in your study, did you include prebiotics and fermented foods as well?
We did, but we didn't push. So we needed to be mindful. It's a pretty rigorous diet, and dietary interventions are notoriously poorly executed by participants, and the data that you get is problematic. I mean, we know that about nutrition studies, that when you use dietary recall, it's like almost useless. It's actually pretty depressing. In fact, let me just say that there's some suggestion that diet, before our study, there was some suggestion that diet might help reverse biological aging, looking at the DNA methylation clock, just mildly. Ours is sort of the first study to really show, hey, if you do it in a concentrated way, you might make a really big difference. But when they looked to see if it made a difference way back and saw a modest difference, they didn't see it in folks who said they ate healthy. They only saw it in people for whom they measured beta carotene and saw that they were eating veggies by blood beta carotene. So that was the marker. So nutrition studies and dietary recall are notoriously poor. So we had to be mindful about how hard we pushed our diet and what our expectations were. And the other thing that we did is we used nutritionists. We've got a large nutrition residency program here, and we've got a team of 4 full nutritionists here who work with our residents, and we accessed our amazing team here to serve as study coaches, just to make sure that people, if they had questions about recipes and so forth, it's not like a normal relationship where you're, encouraging, et cetera. It was a research setting, and so they had a scripted list of questions that they were able to ask only. But within that context, I think the contact with the nutritionists helped people adhere.
Okay.
And normally I think if we had just given people our methylation diet and lifestyle and said, you're on your own, we wouldn't have seen favorable outcome. I think our nutritionists were absolutely essential to the success of it.
Very good. Now, I think I saw somewhere that the overall diet plan is a lower carb and a paleo type diet. Is that correct?
Yeah. So it's a continuum. In our practice, as I said to you, we layer the chief principles, high methyl donor foods and methyl adaptogen foods into all of our diets. But in our study, we kept it sort of paleo, lower glycemic, anti and hypo allergenic. So no gluten, very low low to no legumes, dairy-free.
Okay.
That's what we wanted folks to do. A little bit of time-restricted eating, so nothing after 7, nothing before 7 a.m.
And then you mentioned the exercise that was part of the program as well, and getting enough
Yep.
sleep and stress reduction and everything.
Yeah, so we just tracked sleep. We just had them report how many hours they were sleeping. We weren't sort of pushing a sleep program, but we just wanted them to be mindful of it, and we gave them, sleep hygiene suggestions. Yeah, we used a validated relaxation response program for twice a day for meditation and exercise, whatever they wanted to do, to 60 to 80% perceived exertion.
Did you see changes, improvements in sleep and changes in mood at the end of this
Yes,
program?
we did. I'm so glad that you asked me that. Yes, mood significantly increased in our participants. Yeah, thank you. Minor detail. Yeah, mood increased, cholesterol dropped. What else? We didn't report those in our first study. I think we might have gotten a little nudge up in methionine levels. Folate, as I said, increased, methylated folate. And then we saw our favorable epigenetic changes.
Wonderful. Well, I love that you're so engaged in clinical research, and I really appreciate it. And I always appreciate researchers. So I just want to, give you a shout out for that.
Thank you.
And I saw, when I read your bio, and I went to go and look at one of the organizations that you do some of the research with, Health God Research Institute. So I just wanted you to just tell us what that institute's about, and then there's another one that you work with. Because
okay, sure.
I think it's so important that we're getting so much of this nutrition and methylation research out there. It's very exciting.
Thank you so much, Trudy. Yeah, I feel really blessed to be a clinician and get to engage in research. Very, 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 very blessed. I mean, I was involved in certainly literature research at the lab. I mean, I have, my whole career and published a book of case studies, and so I've been in the research arena for a long time, but to actually get to engage in this clinical study is a dream. And we will continue with a larger population to research our program once we launch the book and so forth. So we'll continue to do this, and just kind of we stuck our toe into the pond with a pilot study, saw some really cool results, and we'll expand it. So Health Gut Research Institute is at my alma mater. It was started by my immunology professor, Heather Zwicky, who's an amazing PhD. She comes from Yale, and so Health Gut Research Institute is at my alma mater, National University of Natural Medicine, and Heather came out, taught immunology. She's an amazing professor, and then concurrently started the Health Gut Research Institute back when I was in school. So it's been around for quite a while, and it has, it's really grown into a premier, natural product, naturopathic intervention, natural product kind of research hub. It's extraordinary, and they do research with the students there. They do industry research. They do studies like mine, and they're just a big, kind of thriving, amazing institute. I'm
Wonderful.
Quite proud of what Heather's been able to create. And my co-PI, Ryan Bradley, is now the director over at Health Gut.
Well, wonderful. I'm so glad we got to share this.
So you know what? Incidentally, any folks working with COVID patients, there's a Health Gut registry where you can upload anonymously outcomes and the interventions that you're using. And so this is global, and you needn't be a naturopathic physician. You can be of any credential, just working with COVID patients. And if you want to anonymously kind of give your experience in treating and what kind of interventions that you're using, Ryan would be most grateful. So giving him a little plug for that.
No, thank you for mentioning that. And I wanted to end with your COVID study. We're in the midst of this COVID pandemic, and you published a paper, evidence.
Actually, 3.
Oh, 3.
It's kind of amazing. Yeah, it's crazy. I didn't anticipate being on 3 peer-reviewed journal articles. That's for sure.
Is there any particular one you'd want to discuss? I was going to discuss. I just wanted you to mention it, and what your findings are, and sort of direction you going?
Yeah, so with Sam Yannick, Joe Pizzorno,
That's the one.
and Helen Messier. Yep. That was looking at understanding that COVID, there are different phases to how we want to address it. We mapped that out in an evidence-informed way using a functional lens, and that's what that paper is about. Sam Yannick led the charge, and the rest of us, contributed extensively, but he did the heavy lifting on actually crafting the manuscript. So I want to give him a big shout out for that. He's a chiropractor. Helen is an MD PhD. Pizzorno is an ND like myself, and we just made this great team, and I just had such a good time working with them, and we've since taken the message to a variety of audiences, and continue to look at the evidence on our phased approach, and it's strong. I mean, we're just getting more and more validated. So phase 1 would obviously be prevention. We know that the comorbidities associated with poor COVID outcome are things that we address in our functional medicine model all of the time. So, anti-inflammatory diet, making sure that we have adequate vitamin D, making sure our zinc stores are replete, the fish oil or SPMs, just making sure all of that is smartly dialed in as a prevention. And then, turning up the volume on certain nutrients to really spike a strong immune response initially. And then if we need to move towards a potent anti-inflammatory response, if somebody is at risk for cytokine storm, jumping on those nutrients. And then the recovery piece, which is huge. We know that the sequela post-COVID can be strong and particularly debilitating. So, various nutrients we've outlined, and acetylcysteine is a big player. We're thinking about glutathione actually coming back to our conversation today as an essential and important piece. So that was the phases paper. If you go to my website on the Vision and Mission tab, you'll see publications and you can access it there.
Let me just go ahead and read the name of that one. It's called Evidence Supporting a Phased Immunophysiological Approach to COVID-19 from Prevention Through Recovery. And then, of course, you can go to Dr. Fitzgerald's website as well.
Yep. The other one that I think is really interesting is the furin cleavage complex. Probably the reason this particular SARS is so virulent and aggressive is because there was this gain of function mutation that prompted the development of this furin cleavage complex. And I've kind of written my thoughts around that and published them. You can read them as a blog on my website in a more plain language, sort of colloquial version, or you can pull the preprint is there now. The publication itself is pending. So there's a couple versions.
Great. Well, thank you. I'll make sure to include those. And I must give a shout out for your blog and your newsletter. It's fantastic. It's
thanks. Thank you so much.
It's one that I always open and I always read. So
that's terrific. Thanks. Yeah, the blog, the newsletter, the team is amazing. The podcast, we've got a lot going on.
Yeah. Go ahead and share your website where folks can find out more about your work.
It's my name. We're very team centric, but I got this website a long time ago. It's just drkarafitzgerald.com. So dr as in Dr. Kara, K-A-R-A, Fitzgerald, Fitzgerald.com.
Perfect. And this has been fantastic. So appreciate you sharing. I appreciate all the work that you do. And I'd love you to just end with some final words of wisdom for us, Dr. Fitzgerald.
So thanks. Okay, good. I think we're in the middle of a revolution in medicine. We are in the omics era. Our ability to see what's going on is more powerful and profound than it's ever been. We're just at the beginning, however, and there's much more to come, and there's a refinement that will be also coming. And so we need to recognize that and be, kind of circumspect or, careful in our interpretation of this revolution. But it's a very exciting time to be a clinician and practicing medicine in this space, particularly in functional or integrative medicine, because we are in this revolution. The interesting paradox about this, the more that we're able to see, the more that we're able to measure the various ohms, the take home is that we see that these simple, very upstream interventions are far more powerful than we really gave them respect for. Even myself, I'm a naturopathic doctor, and I have been in awe as we look at omics data for what we're able to do with these extremely safe upstream interventions. So it makes sense that we evolved with them. So we're in a good time to be clinicians in this space, and I'm just thrilled to be part of the journey.
Wonderful. Going back to the basics. And then learning how important they really are, which is what you're doing with your research, which I think is very exciting. So thanks
it's
again
really exciting.
for being here today, and thank you for everything that you do, Dr. Fitzgerald.
Thank you.
Thank you, everyone, for joining us for another great interview on the Anxiety Summit. Please do join us on other interviews on the summit. This is Trudy signing off.